Good evening. I think I am audible to everyone. So it has been 3 days that I have started this. Sorry for the late due to personal reasons. Couldn't escape those uh, functions. So last time we have discussed about the structure of muscle and the structure of muscle fibers and various proteins involved in muscle contraction like myosin, actin, tropomyosin, and troponin. In addition to this, there are some other proteins which we are going to discuss now. After these proteins are over, I'll be completing the process of muscle contraction based on sliding filament hypothesis. So let me start with the muscle proteins first. The first muscle protein, apart from the other proteins we have discussed before, is dystrophin. Dystrophin is a rod-shaped cytoplasmic protein, and it is a vital part of a protein complex that connects the cytoskeleton of a muscle fiber. If this is the muscle fiber, every cell has cytoskeleton beneath the plasma membrane. Likewise, here will be there is a muscle cytoskeleton of this muscle fiber. So this cytoskeleton of muscle fiber is connected to the extracellular matrix through the cell membrane, and this is called also as costamere, or this complex is also called as dystrophin associated protein complex, in short DAPC. In skeletal muscles and cardiac muscles, dystrophin is part of a group of proteins. That means there is a protein complex where this dystrophin is also a part. All these proteins of this complex work together to strengthen the muscle fibers and protect them from injury as muscles contract and relax. You know that there is a this is called as Duchenne muscular dystrophy. You might have heard about it. Duchenne muscular dystrophy. So here what is happening? Muscle dystrophin protein is said to be not functional. What is the function of this dystrophin protein? It provides strength to the muscle fibers and protects them from injury as muscles undergo contraction and relaxation. How? By connecting along with the other proteins of this complex to the dystrophin associated protein complex to in turn into the cytoskeleton of the muscle fiber. Thus it gives strength. Okay, so in order to give strength, for example, to your heart, to your hand, you need to give a support to this. Okay, someone hits here so that it will not move because you are giving a support on the other side. Likewise, the muscle fiber is also getting support from this dystrophin. Due to a genetic disorder, this dystrophin is not produced in some human beings, and all the muscles they do not get strength from it, so that they get muscles get become weakened. In other words, we can say that the entire body becomes paralyzed, and that this is what we call it as Duchenne muscular dystrophy. And this is one of the proteins of the muscle which is said to be significant uh, after the discovery of this particular disease in the individuals. This is how it looks like. So this is an extract from a particular website. This is the sarcolemma, the muscle plasma membrane, and this is the dystrophin protein, and this is the cytoskeleton, that is actin. And along with the dystrophin associated protein complex, here this protein is a glycoprotein, you can call it as dystrophin associated glycoprotein complex. It is connected to the extracellular matrix, thus gives strength to the entire muscle as it is getting contracted and relaxed continuously during the process of what we call movement or locomotion. And another muscle is said to be the titin. The name itself says it is the largest protein. It is also called as connectin. It is a protein in humans. It is encoded by a gene called as a TTN gene. TTN refers to titin. It is the giant protein, that means high molecular weight protein, and its size is 1 micrometer in length. And what is its function? It functions as a molecular spring for the passive elasticity of the muscle. For example, this is the muscle. There is I band followed by A band, etc., where there are thick filaments and also thin filaments, isn't it? So the thin filaments slide past the thick filaments so that the length of the muscle decreases. This is how it takes place. And during this, what happens? There is every chance for the muscle to contract as long as the H disc is filled by the uh, actin filaments, which we are going to discuss now. During relaxation, it has to come back to the normal position. When you take a rubber band, okay, you what we call stretch it. It stretches. When you release it, it comes back to the normal size. Okay. Likewise, during relaxation, there is every chance for the muscle to go beyond the normal length. So, in order to arrest this, there is a protein. There is a long protein that is tightened, so that during relaxation, the muscle which is decreased to this length should come to the normal length only. If it goes beyond this length, there is overstretching and there will be overload on the muscle. So here this tighten, what does this do? It acts as a molecular spring and is responsible for the passive elasticity of muscle, so that overstretching of the muscle is said to be prevented. So thus, the primary function of this protein is giving elastic stabilization of relative positions of myosin and actin filaments during the process of contraction. And this is how it looks like. So it extends from the M line to the Z disc. So what happens? Overstretching above this limit is not possible. Above this limit is not possible. So that relaxation is within the limits of the titin protein. Okay. And the third protein is said to be desmin. We know cytoskeleton has three different types of proteins. One is called as microfilament, another one is called as microtubule, and the size between these two is occupied by intermediate filaments. We'll deal with later about cytoskeleton. And desmin is the major intermediate filament present in skeletal muscle, cardiac muscle, and also some smooth muscles. And what is its function? Its function is to maintain the cytoskeletal integrity. How? By linking Z bands to the plasma membrane. So these Z bands are linked to the plasma membrane by the desmin proteins. Thus, the cytoplasmic integrity, cytoskeletal integrity is maintained. Okay. And here you can see this. Desmin misfolding, phosphorylation, and degradation is found to cause hemodynamic stress also. So this is the protein. This desmin protein connects the Z disc with the plasma membrane so that the cytoskeletal integrity is said to be maintained. And another important muscle that is generally asked in examinations is, what is the muscle present in the Z line or Z disc? Generally it is Z line, we call it as. Z line is also called as Croce's membrane. A scientist called as Croce discovered it, hence it is called as Croce's membrane. And the protein that is present in the Z line is called as alpha actinin 3. And what is its function? To influence the cohesiveness, togetherness of the cytoskeleton How? By cross-linking actin filaments and other cytoskeleton components so that it creates a scaffold. Scaffold means a surface-like thing that imparts stability and forms a bridge between the cytoskeleton and signaling pathways. So here, this Z line having the actin filaments is said to be the one we distinguish the what we call the structural and functional unit of the muscle as sarcomere. What is sarcomere? It is the entire muscle fiber, region of muscle fiber present between two Z lines. So these Z lines are said to be responsible for holding the actin and myosin filaments during the process of muscle contraction. And that support is provided by the alpha actinin protein present in the Z line. Okay, here it is said to be alpha actinin is present in the Z line and desmin is present at the end of the Z line. In addition to this, there are other proteins, so many proteins like myopodin, which is present between the myosin and the Z line, and there is an actin binding domain, and so many proteins involved in. But actually, we are concerned with myosin, actin, tropomyosin, and troponin only for the process of muscle contraction. And coming to the myosin, it is the first muscle protein discovered by a German scientist called as Willy Kuhne in 1864. In 1939, a Russian husband and wife team, Vladimir Alexandrovich and Miritsa Nikolaevna, discovered that myosin had an enzymatic activity called as ATPase, so that it can break down ATP to release energy. So there are two things with respect to myosin. The first thing is that it is the first muscle protein discovered. Second is that two scientists, Vladimir Alexandrovich and Militsa Lubimova, discovered that myosin has an ATPase activity. When we have studied the myosin, we have seen that myosin has a tail portion and a head portion, and this head portion is said to be having ATPase activity so that it can break down ATP, release ADP, inorganic phosphate, and energy. That was discovered by two scientists. And coming to the discovery that ATP as source for muscle contraction, this is also one of the important bits. The scientist that is involved here in this particular research is Zent Gorgi. S is silent here. Albert Zent Gorgi is a Hungarian physiologist. He turned his focus on muscle physiology after winning the Nobel Prize in Physiology or Medicine in 1937 for his works on vitamin C and fumaric acid. So he worked on vitamin C and fumaric acid, for which he got Nobel Prize. Later he worked on the muscle physiology. In 1942, he demonstrated that ATP was the major source of energy for muscle contraction. That is the path breaking, what we call discovery with respect to muscle contraction. He also demonstrated that actin and myosin are not separate during the process of contraction. They form into actomyosin complex. So this actomyosin complex drives the ATP, is said to be driven by the ATP as the source of energy. So for the actomyosin complex to be formed later, leading to the muscle contraction, it is under the regulation of the energy that is released by the breakdown of ATP according to Zent Gorky. And finally, we come to the sliding filament theory. So this theory is proposed by two scientists, Hansen and Huxley, in 1954. And It was published in the most prestigious Nature journal under the name Structural Changes in Muscle During Contraction. But this theory is not what we call originated all of a sudden. It is said to be there are two scientists who paved the way for the entire sliding filament mechanism. And this group of scientists include firstly Huxley and Neiderger, and secondly Huxley and Hansen. So two hypotheses were put forward at that time. And first coming to the Huxley and Neiderger hypothesis. According to them, the eye bands are composed of actin filaments. We know that thick filaments are said to be restricted to the A band, like these are the thick filaments, and thin filaments are said to be extending from the eye band until the Hansen's disc. That is what we know. So up to this point they extend, not to the Hansen's disc. This is how it looks like, isn't it? And this thin filament is made up of actin filament, tropomyosin and troponin, and most importantly actin filament, which takes part in muscle contraction by forming a complex with myosin. And it is present in the A band. This is A band, this is I band. So actin filaments are said to be present in the I band. Now what happens is the I bands are composed of actin filaments. That is what we have seen here. I bands are composed of actin filaments, and A band comprises only of myosin filaments. So A band we have only myosin filaments, whereas I band contains only actin filaments extending up to the Hansen's disc region. What these scientists Huxley and Neiderger say is, during contraction, the actin filaments, that means the thin filaments, move into the A bands between the myosin filaments. So this is one myosin filament, another myosin filaments, and during the process of contraction, these actin filaments, that means those that are present in the thin filament, they move into the Hansen's disc. They move into the Hansen's disc. That is what their discovery is. During contraction, the actin filaments move into the A bands, where actually Hansen's disc is the place where these thin filaments before contraction are not extending. After contraction, during contraction, they fill the gap in the Hansen's disc. Thus, they move into the A bands between the myosin filaments. That is the first discovery made by Huxley and Neiderger. Later, Huxley and Hansen proposed that, okay, and Huxley here is Hugh Huxley and Hansen is Jean Hansen. They proposed a publication named as Changes in the Cross Striations of Muscle During Contraction and Stretch, that means relaxation, and their structural interpretation. Okay, and their study is based on the rabbit muscle, and they used both phase contrast microscope as well as electron microscope, and it shows the points like this. The backbone of a muscle fiber is actin filaments, which extend from Z line up to the one end of H zone. If this is the Z line, this is the A band, thin filaments, and between the I bands there is A band. In the A band there is only thick filament, and thin filaments extend up to the what we call Hansen's disc only, not beyond the Hansen's disc. Up to Hansen's disc only they extend. That's what they're saying. The backbone of muscle fiber is actin filament. It extends from Z line up to one end of H zone. This is the H zone where there are no thin filaments, only thick filaments. Myosin is said to be present in this region. So it extends up to one end of the H zone, where they are attached to an elastic component which they named it as S filament. According to them, between the Z lines up to the H disc, they are connected to that region by NS filament. Next, myosin filaments extend from one end of the A band through the H zone up to the other end of the A band. So, if this is the A band and myosin filaments are said to be extending from one end to the other end of the A band. Myosin filaments remain in relatively constant length during muscle stretch or contraction. What they say is here the length of the A band remains constant. In other words, the A band is made up of thick filaments. Thick filaments are made up of myosin filaments. That means the length of the myosin filaments remains constant. A band will not change its length. Likewise, myosin filaments length also will not change during the process of contraction. If myosin filaments contract beyond the length of the A band, their ends fold up to form contraction bands. If the myosin filaments contract beyond the length of the A band, if they contract beyond its length, their ends fold up to form contraction bands according to Hansen and Huxley hypothesis. Myosin and actin filaments lie side by side in the A band, and in the absence of ATP, they do not form cross linkages. So, one more discovery is that there is a myosin filament and there is an actin filament, and between these two, there has to be formed a cross bridge by the head of the myosin. So, these cross bridges will be formed only by the breakdown of ATP, without which the cross linkages cannot be formed. That is another discovery made by them. They also say that during relaxation or stretching, only the I bands and H zone increase in length while A bands remain the same. Why? Because during contraction, A remains the same, only the I band changes. Likewise, during relaxation also, A band, the length of the A band remains the same, only the length of the I bands and H zone, which is previously during contraction is closed by the actin filaments, which we are going to discuss now, is said to be increasing in length. That is another discovery made by them. During contraction, they also suggested that actin filaments move into the A bands, actin filaments move into the A bands, and the H zone is said to be filled up so that the length of the I band's length is said to be shortened, and the Z line, which is previously this long, now comes to this much length. Thus, the actin filaments between the two Z lines are pulled into the Hansen's disk, so that Hansen's disk filled up and A band remains the same, only the I bands shorten. And for this entire process, the driving force is the ATP hydrolysis done by the myosin. This is what they have proposed. Okay, and because it is in the form of bullets, you may not be able to understand it. Now I'll explain the entire process in a most easier way. To understand muscle contraction, we should understand the first phenomenon, that is excitation, contraction, coupling. Okay, so there if any question they want to ask with respect to muscle contraction, apart from proteins, apart from scientist names, this is one question they may ask, that is excitation, contraction, coupling. So here excitation of the sarcolemma followed by the contraction of the muscle, both are coupled. So both these are coupled, or that means connected. How these two different phenomena, excitation followed by contraction, how these are connected to each other, what makes them get connected. So let me start with what we call the neuromuscular junction. So these are the terminal buttons which form the presynaptic membrane, and the sarcolemma which forms the synaptic gutter, which we have discussed. So this forms the sarcolemma, subsynaptic and postsynaptic membranes. And during What happens? Motor activity. The motor nerve sends wave of depolarization all along the length of the axon, passes through the telodendrites, reaches the membrane of the Boughton terminaux. And during this, what happens? Acetylcholine is released. Once the acetylcholine is released, the acetylcholine binds with the acetylcholine binding receptors. The binding of the acetylcholine with the acetylcholine binding receptors increases the permeability of the postsynaptic membrane, that is sarcolemma, for sodium ions, so that sodium influx takes place. Once the sodium influx takes place, the first thing that is done is the sarcolemma is depolarized. Okay, this depolarization is nothing but excitation. So, by the chemical transmission, the sarcolemma is depolarized, leading to excitation. That is the first thing. So, how the excitation takes place? So, let me clear this in simple terms. The first thing is depolarization wave of axon reaches the presynaptic membrane, that is membrane of the telodendrites. Now what happens? Acetylcholine is released into the neuromuscular junction. After acetylcholine is released, acetylcholine binds with acetylcholine binding receptors. Once they bind with the acetylcholine binding receptors, the sodium permeability of sarcolemma increases. And the sodium permeability of sarcolemma increases, it results in sodium influx. That means from the extracellular fluid, that is synapse, sodium ions go through the sodium channels of the sarcolemma into the sarcoplasm. And during this process, what happens? Reversal of charges takes place. The reversal of charges leads to the what we call it as depolarization, and this depolarization what we call it as excitation. Thus the wave of depolarization of the axon depolarizes the sarcolemma, leading to the excitation of sarcolemma. That is the first thing. Now it has to undergo contraction. So before contraction happens, the excitation has to be coupled with contraction. How this takes place? So the sarcolemma is excited. Previously there is positivity outside and negativity inside. Now due to depolarization, positivity is inside, negativity is outside. So there is a depolarization of the sarcolemma by the formation of local circuit currents. What happens? The depolarization wave passed on the entire length of the sarcolemma. It passes on until it comes in contact with the T-tubule region. This is the T-tubule region. So the first thing is depolarization of sarcolemma has happened. Now depolarization of the sarcolemma, which is invaginated to form the T-tubule. So the T-tubule is said to be depolarized. And the T-tubules, we know that they are connected to the terminal cisternae of the sarcoplasmic reticulum. So now what happens? The membrane of terminal cisternae of sarcoplasmic reticulum is depolarized. So the depolarization wave by the formation of local circuits of current passes along the length of the sarcolemma, leading to the depolarization of the membrane of the T-tubule, followed by the depolarization of the membrane of the sarcoplasmic reticulum. The main function of sarcoplasmic reticulum, we know that it stores calcium ions, where the calcium ions are bound to a protein called as calsequestrin. The protein is called as calsequestrin. So it is bound to the calcium ions are bound to this protein calsequestrin. When the depolarization wave reaches the sarcoplasmic reticulum, now the calcium channels are opened. Calcium gets separated from the calsequestrin and it is released into the sarcoplasm. Okay, so the next event is release of sarco release of calcium ions from sarcoplasmic reticulum. So what is happening? We see that depolarization of sarcolemma has taken place. It leads to the depolarization of membrane of the T tubule, which leads to the depolarization of terminal cisternae, leading to the depolarization of the entire sarcoplasmic reticular membrane, which opens the calcium ions. Calcium channels are opened, and these calcium channels release calcium into sarcoplasm, which initiates contraction. Okay, so what happened? This entire process is said to be called as excitation, and this excitation is followed by contraction. Between these two, there is release of calcium ions into sarcoplasm. This release of calcium ions into sarcoplasm is what couples excitation and contraction, and this phenomenon we call it as excitation contraction coupling. So in the examination, the question may be like this: excitation contraction coupling is accomplished by, and the answer is the release of calcium ions into sarcoplasm. Okay, excitation followed by release of calcium ions, which leads to contraction. So this is the thing which couples excitation and contraction. Hope you understood up to this point. Now coming to the sarcolemma, inside which now there are calcium ions, and we have dark bands. We have dark bands, that is A band followed by I band on either side. Okay, and the difference being A band is said to be made up of thick filaments, and I band is said to be having a Z line at the center. Stretching between these two Z lines, there is a thin filament made up of actin, which extends only up to one end of the Hensen's disk. This is the Hensen's disk. So Hensen's disk contains only, when you see this, there is only thick filament, whereas the A band contains, that means up to this point it contains thick filament as well as thin filament, whereas the I band contains only thin filament. Now we already have seen that there are thick filaments. Okay, let me explain this again. So this is the thin filament. This thin filament is made up of what? It is made up of actin filament, which is double helical, having a number of active sites, and these active sites are closed by the tropomyosin, which runs to the grooves of the double helix. It covers all the what we call the active sites of actin. Now, in addition to this, there is another protein called as tubulin with three subunits: troponin T, which attaches the troponin complex to tropomyosin. And another one is troponin I, which inhibits contraction. And another one is troponin C, which can bind with calcium ions. So during excitation contraction coupling mechanism, calcium ions are released. And this actin filament is made up of actin. This thin filament is made up of actin followed by tropomyosin, and we have troponin subunits. So the released calcium ion binds with the troponin C. Once it binds with troponin C, the inhibitory action of troponin I is said to be stopped. When the inhibitory action of troponin I is stopped, troponin T, which binds the tropomyosin to actin, is removed. What happens? Troponin C binds, followed by the removal of the tropomyosin. As the tropomyosin is relieved from the active filament, now we see that all the active sites of actin are said to be exposed. Now this entire actin filament is made up, this entire thin filament after the removal of tropomyosin is having only actin filaments where there are a number of active sites being exposed. That is what happens here. Okay, so let me explain you in this way. The first thing is that excitation contraction coupling releases calcium ions into sarcoplasm. Calcium ions bind with troponin C, which eventually stops or inhibits the inhibitory action of troponin I. When the inhibitory action of the troponin I is said to be stopped, troponin T is removed from tropomyosin. When the troponin T is removed from tropomyosin, tropomyosin is lifted off from actin. Once the tropomyosin is lifted off from actin, the active sites of actin are now exposed. So how the contraction is initiated by calcium ions? By exposing the active sites of actin. So far there are so many active sites here in the actin. Okay, all these active sites of actin are said to be hidden by what we call the tropomyosin, and the troponin plays a part in binding the tropomyosin through actin. When calcium ions bind with troponin C, what happens is troponin is removed, tropomyosin is removed, and all the active sites are said to be exposed. Okay, now what happens is after the active sites of actin are exposed, now let me explain the next process. Here there is actin filament, and this actin filament, thick filament. This thick filament is made up of a number of myosin proteins. Now we have actin filament on either side extending up to Hansen's disk region. This is the A band and this is the Hansen's disk. And adjacent to this, there is I band. Now what happened is, as that tropomyosin is removed, all the active sites of actin are exposed. Okay, we have seen that the myosin molecule is having a tail and a head, and the head shows ATPase activity. Okay, so that ATP is broken down to release ADP, inorganic phosphate, and energy. Okay, so act sorry, I removed it totally. So here there is myosin. Just a second, I'll write fast. Okay, and there is actin up to Hansen's disk, and there are active sites of actin being exposed. Now what happened is, myosin head breaks down the ATP. In so doing, it releases ATP. In so doing, it releases energy and ADP. And that ADP molecule is bound to the head region. ATP is broken down to release ADP, inorganic phosphate, and energy. Energy is also used. Before that, what happens? The ADP molecule that is released binds to the head of the myosin molecule. Okay, this is what happens. After that, we have seen that myosin molecule is having two hinges based on which it can bend like our fingers. Our fingers have 1, 2, 3 hinges where the finger can be bent. Likewise, myosin has two hinges based on which it can bend or stretch. So based on the first hinge, the myosin with ADP being bound to the what we call head region extends, it extends and connects to the active site of the actin. So now, sorry, this stretches and binds to the active sites of actin. This is how it stretches and binds. So here also, on the other side also, the same thing happens. The myosin stretches and binds to the active site, and this forms what we call it as a cross linkage or cross bridge. So here a cross bridge is formed based on first hinge when the myosin stretches and attaches to the active sites of actin filament, and thus we come across with the formation of a complex between actin and myosin. We call it as actomyosin complex. So after the active sites of actin are exposed, the next event is formation of actomyosin complex. This is actin, this is myosin. Both are connected by cross bridges forming actomyosin complex. So now what happens? After the formation of actomyosin complex, what happens is, here some amount of energy is released. Based on this amount of energy, based on this amount of some amount of the energy that is released, the myosin, based on its second hinge, it moves into the Hansen's disk. Previously it is like this, now it is moved into the Hansen's disk. As it is moving into the Hansen's disk, what happens? It pulls the actin filament along with it to the Hensen's disk. Previously, this much gap is present. Now, this much gap is said to be closed. Why? Because the actin filaments are brought into the Hensen's disk where there is a space, isn't it? And for this, the Z line with its actin alpha actin 3 protein facilitates as a scaffold, as a structure which supports this pulling. Okay, so thus these are said to be pulled inside, and they are still, this is still attached to the active sites 1 and active sites 2, and there are still more active sites on the actin filament. Now what happens is the same thing happens on this side also. Now what happens is, as this tilting of the myosin filaments into the Hensen's disk takes place, we call this as power stroke. We call this as power stroke. So power stroke is effective. After power stroke means it uses energy that is released during breakdown of ATP by myosin head. And after power stroke is said to be effected, after power stroke is effected, what happens is, previously in the myosin head region, what is there? ATP is broken down to release ADP, and ADP is being attached. Now this ADP is utilized, ADP is utilized to resynthesize ATP. So now myosin head will be having ATP. When it has ADP, it binds to the active sites. When it has ADP, it binds to the active sites. When ATP is resynthesized, it is released. It is released from the active site. Thus, sorry, so it is released from the active site, and followed by utilizing the remaining amount of energy, it comes back to the normal position. It comes back to the normal position, which we call it as recovery stroke. Okay, so in power stroke, it pulls, it is tilted towards the Hensen's disk. In so doing, it pulls the actin filaments into the Hensen's disk. In recovery stroke, it comes back to the normal position. Now, what happens here is, it is exposed to new active sites. Previously, these are the two active sites that are bound to the myosin head. After pulling it, the actin moves in this direction, so that when the myosin relaxes or comes back to its normal position, recovers back, it is exposed to new active sites. Now what happens? The same thing. ATP is broken down, energy is released, ADP is released, ADP binds. As ATP binds, there forms a cross bridge and an actomyosin complex is formed, followed by power stroke, followed by recovery stroke. How long this takes place? How long this takes place? So this is the sarcolemma, and these are the thick filaments, and this is Z line, and these are the thin filaments which are separated from each other by this gap in the A band, that is Hensen's disk. During the power stroke and operation of power stroke and recovery stroke continuously, what happens is, the length of the A band decreases. Why? Sorry, the length of the I band decreases. Why? Because the thin filaments, that is actin filaments, are brought into the Hensen's disk, so that what happened previously, this is the length of the muscle which is decreased to this length, and this we call it as decrease in length as shortening or contraction of muscle. So contraction of muscle takes place. How it takes place? It takes place by the attachment followed by detachment of the myosin filament, during which the actin filaments slide past the myosin filament into the Hensen's disc. As it is continuously repeated, it is repeated until the actin filaments are closing the gap in the Hensen's disc, during the activity of which the Z line is pulled inside. Thus, the length of the I band decreases, whereas the length of the A band remains the same. This is what sliding filament hypothesis is. So the name says that the actin filaments slide past the myosin filament into the Hensen's disc, leading to the shortening of the length of the muscle, leading to finally the contraction of the muscle. And all these events are reversed during what we call it as muscle relaxation. Okay, we need not tell what is happening during muscle relaxation. The same events are repeated, and the continuous or alternate power stroke and reverse stroke are said to be explained by two theories, which is called as ratchet theory or walk along theory, both of which suggest that the actin filaments are walking on the myosin filaments into the Hensen's disc. How? By the repeated power and recovery strokes, which is called as the ratchet action. So this is what we call it as ratchet theory or walk along theory, which explains us the sliding movement of the actin filaments over the myosin filaments. Okay, to sum up this entire thing in few points. First of all, what has happened? Excitation, contraction, coupling has taken place. How? By the release of calcium ions. Then what happened? Calcium ions are bound to troponin C. Then inhibitory action of troponin I is inhibited, followed by release of tropomyosin from actin, and this is followed by the exposure of active sites of actin. So in the first event, the active sites of actin filament are exposed. After the active sites of actin filament are exposed, the second event is formation of actomyosin complex. How it takes place? The myosin head is involved in breakdown of ATP, releasing ADP, inorganic phosphate, and energy. And in so doing, the ADP binds to the myosin head. As it binds to myosin head, it stretches and attaches to the active sites of actin. So, binding of myosin head to active sites of actin takes place, leading to the formation of actomyosin complex. So this is the second event. And the third event is walk along hypothesis or ratchet theory, or we can call it as sliding mechanism. And in sliding mechanism, what is happening? The first thing is power stroke. Based on the energy that is released during the breakdown of ATP by myosin head, the myosin head tilts towards the Hensen's disc. It tilts towards the Hensen's disc with the help of the cross bridge formed between the actin and myosin during the formation of actomyosin complex, leading to what we call it as power stroke. After power stroke is effected, the actin filaments move into the Hensel's disc. After the actin filaments move into the Hensel's disc, what is happening? Now ATP resynthesized, and the myosin head is having now ATP. Once the myosin head is said to be having ATP, myosin head is released from actin. So previously actomyosin complex is formed. Now it is released after power stroke. Once the release takes place, it exhibits recovery stroke. Where previously it is like this, now it comes to the normal position, during which now it is exposed to new active sites. Okay, it is exposed to new active sites of actin filament, and the same power stroke and recovery stroke and all these events are repeated until the all the thin filaments slide past the thick filaments and fill the Hansen's disc gap, so that the I band also, the Z line also is pulled inside, the I band decreases, and the A band remains the same during the entire process. So this sliding past of the actin filament over the myosin filament into the Hansen's disc, filling the gap in the Hansen's disc where I band is shortened and A band remains the same, is what we call it as muscle contraction. Yes, this is all about the sliding filament hypothesis. Okay, so any doubts you can ask me, or otherwise I'll ask you one by one stepwise and we can finish this within 5 minutes. Any doubt? Okay, let me start with excitation contraction coupling. Who is going to tell me what excitation contraction coupling is? How it takes place?
Alice takes place by uh, moving of sodium ions inside
Very good. That is excitation.
depolarization moving. Yeah,
So depolarization wave opens the sodium
sodium
channel.
ion
Sodium
inside.
influx takes place leading to depolarization of sarcolemma. That is what is excitation. The depolarization of sarcolemma spreads up to what point?
T-tubule of the sarcoplasmic reticulum.
It leads through the triad. That means tubule and two terminal cisternae until it reaches the cisternae of the sarcoplasmic reticulum. So after the membrane of the sarcoplasmic reticulum is depolarized, what happens?
It releases the calcium ions into the sarcoplasmic. Calcium
It releases calcium ions into sarcoplasm, and this calcium ions release leads to contraction. It initiates contraction. So excitation followed by contraction is coupled with release of calcium ions. Next, what happens after calcium ions are released?
The calcium ions are binding on the troponin TNC part of that.
Very good. Calcium ions bind with troponin C only, not TNC. It binds with troponin C. So the troponin I is inhibited, troponin T is removed. It leads to what? The
It leads to
tropomyosin
lift the tropomyosin on the actin proteins.
lifting of tropomyosin from actin. Then what it leads to again?
Active sites are exposed.
Very good, excellent. Active sites of actin are exposed. So this is the second event. Release of calcium ions followed by exposure of active sites of actin. Then what happens?
The myosin filaments ATP is
breakdowns and forms
the head of the
ADP.
myosin thick filament ATP is active.
ADP
Myosin head breaks down ATP to release energy, in turn where this ADP is bound to myosin head. Then what happens?
Actinomycin complex is bound.
Myosin head will fuse with that
It
actin
binds
sites.
the active sites of the actin protein.
So it binds to the active sites of actin protein, forming cross bridge. This is the cross bridge and leading to the formation of actomyosin complex. Then after formation of actomyosin complex, what happens?
Power
When
stroke.
ATP it stretches into the towards the
Henson.
Henson disk, sir.
Very good. So ratchet action takes place, power stroke is affected, and actin filaments slide past the myosin filament. That is what you have to remember. Actin filament slides past the myosin filament into Hansen's disk, followed by ATP resynthesis, release of myosin head from active site, and again recovery stroke. Again it is exposed to new reactive site, and the same process continues until H disk is said to be closed. Then what is the end result? During muscle contraction, I band remains same in length.
Decreases
A
the
band,
size.
The
Maybe
length of the high band decreases, sir.
Hybrid decreases.
very good. I band does not remain same. The length of the I band decreases, the length of the A band remains the same. That is what Hansen and Huxley explained to us. So, when we were studying these points, you might not be able to understand. What they say, during contraction, actin filaments move into the A bands and H zone is filled up. Only the I bands shorten. The Z line comes in contact with the A bands. Previously, this is the Z line, this is the A band. So, there is a gap between these two. Now they are pulled up to this point. So, the IZ lines come in contact with the A band. Here, only the I bands and H zone increase in length during stretching. In other words, they decrease in length during contraction, whereas A bands remains the same during contraction as well as relaxation. So this is all about sliding filament hypothesis, and with this we come to the end of this particular muscle contraction, muscle proteins, structure of muscle and myofibril. And only we have only these two topics in CUET syllabus. After the life sciences syllabus is over, I'll continue with the remaining physiology topics. Okay, and now tomorrow I'll start with